Our first speaker this evening will be Dr. Natalie Katz from Duke University. Dr. Katz is currently our NMSG Fellow, and she's here to update us on her work. And her topic will be development of novel imaging biomarkers for use in pediatric FSH muscular dystrophy.
All right, so thank you for that introduction. So we'll go ahead and get started. Here are my disclosures. And so really, I know most of you are familiar with FSHD, but I thought I would just start with saying what we know about pediatric FSHD, and most of what we know comes from studying the early onset group. And so from these studies, we know that children with early onset FSHD tend to have a more severe disease phenotype. They have at times been associated with a faster disease progression, smaller D4Z4 repeats, and then we see a higher incidence of extramuscular manifestation in these children. But with these studies, really there is a lack of longitudinal data, and most of these studies are limited by a single center or only a few centers, and they have small sample sizes. And a lot of these studies are actually completed when these children are adults, and they're trying to draw conclusions about disease progression based on how they presented as children and what they look like now that they are adults. So really there's a lack of prospective data in pediatric FSHD. Right now there's only one natural history study that's been published. This was by the Dutch group. And they have this 2-year follow-up study that was published a few years ago. And for those of you who are at IRC, they discussed their 5-year follow-up data, which should be coming soon. But currently, as it stands, this is the only publication that exists that has prospective data on disease progression in pediatric FSHD. So they followed 20 children ranging in age from 2 to 17. About half of them met clinical criteria for FSHD, and for those who aren't familiar or maybe need a refresher, the current clinical definition of early onset FSHD is onset of facial weakness before age 5 or an onset of shoulder weakness before age 10. And it's just a clinical diagnosis or clinical criteria that we use to designate early onset. And over the course of this 24 months, they saw that in this population, the symptoms are actually slowly progressive. And they actually saw an increase in the functional exercise capacity as measured by the 6-minute walk test. And so this is just some of their data. They looked at the FSHD clinical score. The age is on the x-axis, the clinical score on the y. The early onset kids are in the light blue, and what they called the classic onset or the more traditional onset FSHD in the dark blue. And you can see a separation. But when they looked at the clinical score over time, the change in the clinical score over time, they really didn't see a difference in the rate of progression between these two groups. And they also looked at the 6-minute walk test, and as I alluded to, they actually saw an improvement in the function over time. Some of this may be because they're children, and there's a little bit of a learning effect and some behavioral components at times. But they saw an actual improvement in their functional outcomes when they measured it this way. And then they also used neuromuscular ultrasound to look at muscle echogenicity over time. And so with the ultrasound, they did see that the children who met criteria for early onset actually had a higher echogenicity at baseline, and that this echogenicity increased at a higher rate than those who had the traditional onset over the course of the 24 months. And so just when they looked at the early onset between the classic onset children, shorter D4Z4 repeats, younger age at onset, they had a higher age-adjusted clinical score, but again, no significant differences between the rates of the disease progression as measured by those clinical severity scores. And when they looked at a battery of different functional assessments, there really wasn't any difference that they could detect between the two groups. They saw some of these differences on imaging, and then they also looked at the incidence of extramuscular manifestations that was relatively similar across the groups, with some developing facial weakness over the 24 months, and then hearing loss was only seen in those who met the early onset criteria. So here in the United States, we have the ongoing MOVE FSHD study, as many of you are aware of, and I guess it's international as well, but This study is the follow-up study to Resolve, which was really meant to be an all-comer study and to follow patients over several years to see how their symptoms progress and what functional measures are best for measuring those changes. And this study, importantly, does include pediatric subjects. It also includes non-ambulatory individuals, which are typically not included particularly in clinical trials, and it also is including individuals with FSHD type 1 and type 2. And then there's a sub-study to MOVE called the MOVE Plus study, where they're using whole body MRI to guide muscle biopsy in a subset of these individuals. And this sub-study is not open to pediatric subjects. And that's where my ears perked up. And for those of you who know me, I'm always thinking about the kids. And so that's really where the idea for my project came in, is can we also use imaging to look at changes in muscle composition in these pediatric subjects as a potential biomarker for disease progression. And so the fellowship that I was awarded was a pilot study using whole body MRI and neuromuscular ultrasound to characterize muscle involvement and changes in composition over 12 months. And this really was meant to be paired with the clinical and functional assessments that are being collected as part of the MOVE FSHD study. So right now, as it stands, we have 23 participants in the MOVE study who are pediatric subjects, 9 females, 14 males. 16 of them have completed their 24-month assessments, and 3 have actually already completed their 24-month visit. So I think this is a great sign that these families are interested and willing to participate in these long-duration studies. So I wish I had some imaging data to share with you, but as many of you know, when you move institutions and try and start a very complicated study from scratch, it takes a long time, and so I am still waiting to get that up and running at Duke, but it should be happening very soon, and so hopefully next year I will actually have some imaging data to share with you. But we do have some of the functional data. These are the participants broken down by sex. Most of them are Caucasian and of non-Hispanic ethnicity. Their ages at enrollment were fairly similar, and we actually see a fairly widespread in terms of the ages of enrollment from fairly young to adolescents, and some of these kids now are not technically kids anymore. They have reached the age of 18, so I guess we call them adults, but I call them my kids who are not kids anymore. And they have similar functional scales at baseline when you look at it using both the clinical severity score and the FSHD clinical score. Most of them were actually ambulatory when they enrolled. There was one male participant who was non-ambulatory at enrollment, and there was one female participant who actually went from being ambulatory to non-ambulatory during the course of the study. And this is just looking at their clinical severity over time. So the males are in green, the females are in the dark blue or purple. And so it's kind of variable, maybe not much change has been detected using the clinical severity rating scale. We also use the FSHD clinical scale, and so you maybe start to see a little bit more movement as these participants continue on throughout the course of the study. It's still pretty variable and doesn't always detect change year over year. But it's at least a start. So there's lots of other data that I didn't have time to go through, but while this was ongoing, some of you may have heard, and for those of you who don't, my mentor, Dr. Statland, and others, many of whom are in this room, have actually received funding for a U01 to validate outcome measures in the pediatric FSHD population, and so this is being called the MOVE FSHD PEDS study, or MOVE PEDS, because that's not complicated at all. But we're very excited that this was awarded by the NIH. And so the goal is to enroll 80 children across the United States, and then with our collaborators also in Australia who have done a lot of work in the pediatric FSHD space as well. We are anticipating that about 50% of these children will meet the clinical criteria for early onset, but also will give us a better gauge of how do we best characterize early onset FSHD, and is that really the best definition? This study is a little bit different than MOVE. We're going to do the assessments at every 6 months, in keeping with the idea that if they're progressing faster, do the the increased number of assessments help us capture that change over time. And so the aims are really looking at validating outcome measures in preparation for clinical trials using the clinical and functional outcomes with the FSHD-COM as well as reachable workspace, and then also looking at imaging assessments and using quantitative muscle fat fraction on MRI as a biomarker for disease progression. And this is just a schedule of assessments very similar to what we're doing in MOVE. And so with the help of our colleagues in Australia, they have done a lot of work already in the pediatric FSHD space. And so this is from Dr. Ian Woodcock's group, and they have successfully scanned a number of pediatric subjects. And so what I hope you can appreciate is that, number one, they can successfully do this in a child repeatedly. I don't know why they needed so many at the age of 7, but as time goes on and this child ages, you can see, particularly in the left leg and the posterior compartment, that there's significant muscle involvement over time, both in atrophy and fatty replacement. And so this is really one of the first glimpses that we've had into muscle composition in the children. And so it's really exciting to see that we can detect this change over time. And so in partnership with Springbok Analytics, we are taking this raw MRI data and using it to reconstruct 3D images of both the muscle shape, volume, and then also quantifying muscle fat fraction. And so in these images, yellow means lower fat fraction, and red is actually indicative of higher fat fraction, and you can appreciate some of the asymmetry that is typical of FSHD, and then they can also start to use this information to quantify the amount of fat fraction that's present in the muscles themselves, and they've started to do this longitudinally. And so collectively, we're hoping that this will help inform disease progression and provide more information about how this changes over time, and can we use this as a biomarker for disease progression in response to therapy for clinical trials. So hopefully my pilot project will be starting soon in parallel with the MOVE study. The UO1 is planned, and we'll be getting off the ground as fast as we can do it. Maybe by the end of the year, maybe early next year before we're actually able to start enrolling for the UO1 study, but that is on the horizon. As a follow-up to this study, this fellowship, I have submitted a K-12 application to continue with this work in the pediatric FSHD space, with plans to continue on with this research through K-23s and subsequent awards. I also have an interest in looking at other aspects of FSHD, so I'll be submitting a grant soon looking at using long-read sequencing to look at D4Z4 repeat sizes and methylation in the pediatric subjects, and can we learn anything about some of the variation in onset and severity from a genetic standpoint. And the hope is that we can, that I can eventually use these skills from this fellowship to expand imaging and some of these other techniques to other pediatric neuromuscular diseases. So with that, lots of acknowledgments in the room, particularly the neuromuscular study group. Thank you for the opportunity in awarding me this fellowship. My mentor, Dr. Jeff Statland at the University of Rochester, or at the University of Kansas, as well as Dr. Twiel, who's no longer here, but was a mentor at the University of Rochester. My new colleagues at Duke University, as well as our colleagues in Australia and Springbok Analytics. And then finally, I would be remiss if I didn't wish Dr. Statland a happy birthday. He is, you do not want me to sing into a microphone, but he has been a wonderful mentor and given me many opportunities, and I'm just very grateful for the opportunities that he's provided me and many others in this room. So thank you for that, and with that, I'll take any questions. Oh, we don't have to.
Our U project that Jeff has.
Yep. So we have been pretty careful in terms of crafting these different studies so that all of the data is collected in a similar manner across the different sites so that all of it can be aggregated. And so the hope is that we can do that both across the different studies and different sites.
Own MRIs?
Yes, every site will do their own MRIs. They'll all be validated before we start. Every site will run the same protocol so that they can all of the data can be aggregated.
That's always tricky.
It is tricky.
So these kids do have extra motor, extra muscle manifestation, right? That's
Yes.
we're talking about this quite a bit during the meeting.
Yep.
So is there any plan to capture any of those other phenotypical features that are beyond the muscle?
Yeah, those will be collected as part of the MOVE study. We'll be looking at those. And so I don't have those as aims or objectives in this particular study, but certainly something that we're interested in following over time.
And they're going to be incorporated in the UO1?
Yeah, a lot of them are collected as part of the general MOVE, and then yes, as part of the UO1. Thank you.
Liz, you're going to have to tell me when I can, when the slide's fixed. Go ahead. All right. Well, truly one of the highlights of our annual meeting is the Griggs Lectureship. You saw this slide earlier today, sort of chronicling the history of this lecture series and some of the outstanding presentations we've had throughout the years. Of course, the intent of this lecture series is to honor Dr. Griggs for his considerable contributions to the field of neuromuscular medicine, both as a clinician scientist as well as a mentor to countless junior faculty. As you know, the NMSG was founded through Dr. Griggs's efforts, and through the years, he has remained the soul and principal engine of this organization. So it's very, very fitting to honor Dr. Griggs in this way. So, little help? So it is truly a great pleasure for me to be able to introduce this year's keynote speaker. I have known Merit for probably about 25 years and have had the privilege of working alongside her for the last 18 of those years in the same department. Merit, if I remember correctly, was born in Oak Ridge, Tennessee. Has I got that right? Okay. But spent a lot of her early years in Buffalo, New York, which is roughly a 6 to 7 hour drive northwest of where we are currently. She completed her undergraduate degree in chemical engineering at the Massachusetts Institute of Technology and subsequently her medical degree within the Health Science and Technology program at Harvard Medical School. She pursued neurology training and fellowship training at the Massachusetts General Hospital in Boston, and along the way completed a master's degree in clinical epidemiology at the Harvard School of Public Health. Subsequently, Merit remained at Mass General Hospital for her entire career, where she has devoted her efforts to the care and treatment of patients with ALS. About 30 years ago, she doesn't look that old, but about 30 years ago, she established the Clinical Trials Unit at Mass General, and along with Bob Brown, built the leading ALS therapeutic development program in the United States, if not the world. The center is now known as the Neurological Clinical Research Institute. This institute has grown throughout the years and currently employs roughly, I think, close to 100 full-time staff who coordinate and perform data management for trials in several neurological disorders. Outside of MGH, in 1995, she founded the Northeast ALS Consortium and served as the co-chair of this group for the first 20 years or so. This consortium has grown from a handful of sites that were sort of clustered in the New England geographical region to its current state of approximately 150 sites that are spread throughout the United States, Canada, and Europe. The mission of this consortium is to conduct collaborative academic-led clinical trials and research in ALS. She's also the principal investigator for the Clinical Coordination Center of Neuronext. She's won over 20 awards, including the Sheila Essay ALS Award from the American Academy of Neurology, the Forbes Norris Award from the International Motor Neuron Disease Alliance, the Pinnacle Award from the Boston Chamber of Commerce, the Ray Adams Award from the American Neurological Association, and last year, the MDA Legacy Award for Achievement in Clinical Research, marking the third time she's been recognized by the MDA with an award. She's published over 300 articles and along the way has mentored numerous physician scientists in the field of ALS. Her current titles include the director of the Sean Healy and AMG Center for ALS. She's chief of neurology at Massachusetts General Hospital, and she's the Julianne Dorn Professor of Neurology at Harvard Medical School. And tonight she will be speaking to us on therapeutic developments, both in trial structure and drugs in ALS.
Thank you, Bill, for those really kind words. And thank you for inviting me to be here at your meeting and for this great honor. I wanted to take a few moments to remember the way that, Birch, you've had an impact on my life and my career. When I was a resident, Ann Young, who was my chair, I told her I wanted to do clinical trials, and she said, well, go over to Rochester and learn everything that they're doing there, because they know how to do this. And Ira Schulzen and Carl Kuberts and the entire team that worked for you were so generous in just sharing what they had built in Parkinson's and Huntington's, and I learned so much from what they did. And they, Ira, recently passed, but till, just a couple months before, he was still giving mentorship. The other thing, I met you very early on, you might not remember, but at the Academy of Neurology, when I got one of the early grants from that Natalie has just received, something like that. And you and Bud Rowland would take the awardees and introduce us to other people at the AAN and really help mentor young people. And I learned quickly that if you called me, it was usually to ask me to do something. So that's one thing. But, and you can't say no to Birch. But the other thing is that usually it was something actually that ended up being incredibly helpful, to be on different committees at the AAN and to meet other people and really to build that network that you need to really succeed. But one of the memories I wanted to really share, which is, you guys probably don't know why there's a treadmill on my picture, but it was because for one of my very first grants that for a trial with ceftriaxone and ALS, I had to go to the NIH for the review. That's when they used to make you come and present to the committee in person. And I was so nervous about this. So I went down to the gym to try to get off some of my energy, and I'm not an athlete, but there was an athlete there. That was Birch, running so fast and sweating at 6 a.m. I knew he was my reviewer. I said, okay, I'm not doing this, and I ran out the door. But anyway, thank you for everything you've done for my career and for so many people in this room, and this is really quite an honor. Thank you. So today, first, these are my disclosures of grants and advisory boards. And again, I'm on some of those American Brain Foundation and boards because of Birch. So this is just an incredible time in neuromuscular. We're seeing so much excitement in new therapies, but in ALS, it's also moving at a very fast pace. It's really quite exciting to see. We heard a great talk earlier today by Pietro about some of the biology, the TDP-43 biology. He set this talk up very well because a lot of what he described is leading to therapeutic interventions in people living with ALS. We do have some marketed drugs that have some efficacy, but we have a lot to do still. We have two drugs that are for all forms of ALS, riluzole and radicava, that modestly slow progression. We have a new gene therapy drug, which I'll show you much more about, that is really transformational for the field, really telling us it is possible to treat this illness if you can get close to the cause of the illness. And we have multidisciplinary clinics that really make a big difference for people and their families. This ALS in the last couple of years has really become a disease where the patients and the families have become very strong advocates. And this was not always the case, but I think between social media and some really key patient leaders, they are really partnering with all of us to try to cure this illness, and they're advocating for more research. And in fact, recently got a bill passed by Congress for $100 million a year for ALS research for 5 years. So it makes it even more fun to be in the field, to be working so closely together. There's also global networks that work together. Bill mentioned NEALS that we started really in the spirit of the Parkinson Study Group and the Huntington Study Group, but there's a European one, NCALS, and a Pan-Asia one, PACT ALS, and these groups get together often to share kind of best practices around trials. And there's a lot of companies in the space. So, that was kind of one of the reasons that led to our platform trial that I'll tell you about. But it, again, gives hope for people living with ALS that we might make some real impact. The Northeast ALS Consortium is no longer in the Northeast, but is really a great group, like this group of people who care about the same disease coming together. We have 154 centers. Anybody at the center is part of NEALS, so it's very multidisciplinary. We've run a lot of studies and really have the kind of mission that this group is about kind of speeding drug development. So we share all the data from our trials on an open forum called PROAC. All the samples from the trials are shared. We set up some infrastructure to kind of speed up trials like single IRBs, and we do a lot of training. And there's a great science advisory board led by Tim Miller and Bob Brown and others that will help academics as well as industry on really any part of the drug development program that they need help on. We've had a succession of leaders, and it's currently led by Jinsey Andrews at Columbia and James Berry from Mass General. So today I wanted to focus just on 2 topics so we could really, one, I didn't know how long I had to talk, so, but you could talk about a lot more, but I thought these are the exciting areas in ALS therapeutics. And so to talk about some of the antisense oligo approaches, as well as some other gene-targeted approaches that are coming for not just the genetic form of ALS, but also for the more broader form, and then tell you a little bit about the, where we are on the Healy ALS platform trial. So when we see patients, about 10% of them will have multiple members in the family with ALS, and 90% will only have one person. But the field's really moved away from thinking that there's familial and sporadic, because based on some particularly twin studies and other kind of large cohort studies, we do think that there's a fair amount of genetic risk factor in the singletons, and that maybe about 50% of the risk might be from genetic changes. But the discovery of the genes within those 10% have led to a lot of models of the disease as well as understandings of biology. And this is a slide I borrowed from Sammy Barmuda in Michigan, but really trying to group the different gene mutations in different themes of biology to help us with, again, therapy development. And so the big areas are many genes in protein turnover and RNA metabolism, axonal transport, and mitochondrial function. So there's been a lot of trials now targeting different genetic forms of the illness, and I'm going to give you the more details of the ones in red, but I did want to point out that just like in SMA, that once you had one therapy that came forward, that other interventions came to try to add to it or improve on the method. And so our first ASO treatment for SOD1 ALS is called Qalsody or tofersen, and that is a marketed drug. But there are now at least 3 other groups, if not more, working on other ways to shut down SOD1. So SOD1 was the first gene that was found to cause the familial form of ALS, and lots of science suggests that if you can lower the amount of the protein, that can be efficacious, and that's what the ASO does. So now there's company Unicure, that is currently enrolling patients in an SOD1 trial using an AAV10 capsid that has an siRNA that blocks SOD1. And so they're enrolling currently in the United States at about 11 centers. So that's a one-shot treatment. It's challenging, just like an SMA, of how you do these trials when you have a marketed drug that has a lot of efficacy as well. Regeneron and Alnylam are planning in the next couple months also a trial in SOD1 with a C16 bound, a conjugate to siRNA that has to be given intrathecally, but the advantage here could be that it could be given less frequently than the Qalsody one because of its half-life. And then Voyager is working on an intravenous way using their vectorized capsid that can get across the blood-brain barrier to also knock down SOD1. So for a form of ALS that's very rare, there might be about 300 new diagnoses a year in the United States. There's a lot of activity to try to get this to the point where it's easier for patients, whether that's going to be a single IV or a single intrathecal injection. So very, very exciting. There's other genes that people are targeting, and I'll show you some work by Dr. Neil Schneider at Columbia, who developed an ASO for another form of familial ALS caused by mutations in FUS. And then, as we heard earlier this morning, there's efforts to try to target UNC13. The C9orf72, which is the most common cause of familial ALS, has been more difficult to kind of target, but lots of groups trying to do that. There have been 2 ASO trials that were both negative. But like other triplet repeat disorders, the gene therapy approaches still haven't quite been figured out. And then, sometimes our patients ask us why, all this work is happening in, 10% of the population, not the rest. And, first of all, that's not true, but the second is that the technologies that are being used to shut down the genes that cause ALS are also being used to target different proteins of interest in the broader population. So I'll show you at least the design of a current trial that's enrolling patients with an ASO that will help make full-length Stathman-2. So we heard this morning that one of the problems with when TDP-43 is mislocated is it can cause these cryptic exons or missplicing, and so one of the things that does is cause a shortened form of Stathman-2 and axonal degeneration, and replacing full length can be, at least in animal and cell models, can be efficacious. So that is currently enrolling patients. And then there's other things coming. There's ways to target TDP-43 using AAVs to deliver antibodies intracellularly to break up TDP-43, and that's being developed by Vectory. So lots of exciting things, not just for the genetic form, but also for the broader singleton populations. So that's a lot of activity, just using different gene modification approaches. So I'll go through some of the ones in red here. So I mentioned the SOD1. So while it was the first one to be found, it's probably the second most common cause. So it causes about 1% to 2% of ALS, and there's about 350 people living in the United States with that. This trial was published in 2022, and Tim Miller is the lead investigator of that, really a global study to treat this form of the illness with an antisense oligo called tofersen or Qalsody. And some of you might have seen this data. It's been presented by Tim and others, but I want to just give the highlights of the trial. This is a global trial that it really was a phase 1 to phase 3, adapted along the way, but I'll show you the phase 3 part. Enrolled about 100 patients, 72 on active drug, 36 on placebo. There was a loading period, an intrathecal injection, and then monthly infusions for 6 months, and that was a double-blind period, and then everybody was put on active drug. And in SOD1, there's over 100 different point mutations, and some are associated with very fast course, some kind of middle, some very slow. And so the primary outcome measure was in the fast progressor mutations. So first thing is did hit the target. It lowered SOD1 protein in the cerebrospinal fluid. So here, blue is the group that received placebo first, and then red is the group that got active drug first. And you see that it lowered it about 40%. And they knew from animal studies that this amount of CSF lowering was about equivalent to 80 to 90% lowering in the spinal cord. It also lowered this biomarker of axonal injury, neurofilament, and this change was actually what led to the approval. It was the first drug in ALS approved on an accelerated basis based on a biomarker change, and that, besides being very important for this drug, is very important for the field and has gotten people excited that there might be a way to get drugs approved faster if they lower this particular biomarker. So by 12 weeks, we saw about a 50% drop in the active group. And then when people switched from early, for who are on placebo, to active drug, that also lowered in that time frame. So the primary outcome measure was this ALS functional rating scale, which is a scale from 0 to 48 that typically drops 1 point a month. In people with the fastest mutations in SOD1, it can be much faster than that. The placebo group, again, is shown here in blue and the active in red. And we didn't meet the primary outcome measure. As happens in many trials, the groups don't exactly do what you thought they were going to do. So the placebo group didn't actually decline as fast as expected in this group. However, we learned a lot from the open label extension. So people remained blinded to initial treatment assignment when they went into the open label. And they're still ongoing. This study, people are still in the open label extension, not knowing what they were assigned to during the double blind, but that's important kind of long-term information to have. But what we started to see is a bigger separation in the group in that active treatment stage. And with the group that was assigned early to placebo, never really catching up to the group that started on active drug first. And we saw that in all the measures, in ALSFRS and breathing. And then we also saw it pretty dramatically in the strength measures with really kind of a flattening of the curve after a couple months in the group that got the drug first. So we learned a couple things. One is that the study was underpowered. If we had more patients, we probably would have had a statistically significant on the primary, but more importantly, it actually took longer than 6 months for this drug to work, even though to clinically work, even though we saw the biomarker change early. So we also looked at whether some people improved, because we were really hearing from sites, from the physicians, that they were seeing some of their patients stop progressing and some of them get better. And we don't usually hear that from our sites. And so in fact, about at the best group, the 40% improved. These were people who started the drug earlier and who were, based on their neurofilament baseline level, in a kind of what we call a slower progressing group. That's kind of in the bottom here. Anyway, super exciting. There's we didn't know that this was actually possible in the illness. Now, of course, it's not everybody, and it's not just getting in there early, so there's probably other reasons why some people responded better than others. But it's the first time in the field of ALS that we've seen anyone stop progressing or anyone get better. So it's kind of reinvigorated in a way the whole field. We also saw a benefit in survival. Now, the group with mutations there were in the slower progressing group, again, here defined by their neurofilament level. Most of them, fortunately, survived the study. But when we looked at the faster progressors compared, again, to early treatment versus starting later, there's about a 1.6-year extension on survival. So this drug is now a marketed drug since April of 2023. A lot of the clinics are administering this. It is a monthly intrathecal injection. It's largely safe. There are some risks. There's some increase in CSF protein levels. There were about 4 people who developed transverse myelitis, but it can be something that you treat through with steroids, and people do want to stay on the drug. So we saw this lowering of SOD1, we saw the lowering of neurofilament, and that predicted the responders. We saw the prolongation of survival, and again, some people stabilizing, some people getting better. So that study then led to this study that's ongoing, which is a prevention design trial for people who are gene carriers for SOD1. This study is being led by Dr. Michael Benatar from University of Miami. And this is a very creative design. And there's two things. One is we want to know whether early intervention will prevent the onset, but this is also a regulatory requirement because the drug was approved on an accelerated basis, conditional on this second study happening. So in this study, people who carry SOD1 genes and are asymptomatic are enrolled. They're followed periodically with exams as well as neurofilament levels. When their neurofilament level goes up to a certain amount or a certain percent, they're randomized to active drug or placebo. And they're continued in that until they might get symptomatic. There is an out for anyone who's symptomatic goes on open label to tofersen drug. So this is actively enrolling. And it's a global study, and I don't think it's that far from being completed with enrollment, but if you have any families with SOD1, it's definitely looking for some more participants. But in the ALS field, this is another milestone. It's the first time we're looking at prevention studies, and it's really couldn't have happened without a lot of the work that Michael Benatar and others did to follow people in a natural history study who are gene carriers to know how to determine what happens before onset. And he had shown that about a year before clinical onset, neurofilament would go up in these patients who are gene carriers. So now I'm going to switch to the second ASO study, and this is in FUS. And again, this work is really pioneered and led by Neil Schneider at Columbia University. So FUS is happens about 3% of families, so it's not a very common mutation, but it's a very aggressive mutation. It happens in all ages, but it happens in also juvenile onset ALS, and the younger the onset, the faster the progression. Neil really used the same technology that was used in the SOD1, same type of ASO approach, and he started with single patient expanded access protocols, and once he got up to 10 or 11 patients, the FDA did say now it's time to do a study on it rather than single patients. But in those single patient studies, he was seeing some people get better, really some remarkable improvements, especially in the juvenile form, and reduction in neurofilament. So people are very excited about this. And there is an ongoing phase 3 global study called Fusion that he's leading with Ionis and several other people. And in this study, people are randomized to active drug or placebo, 2-to-1. It is a long study, 18 months, and that was really based on his data in the compassionate use that showed that it actually took that long for at least one of the patients who got remarkably better for her to start to really improve. So it's a long study, but there is an out. If patients get worse by a certain amount, they're taken out of the study and put on the active medications. So this is also going to be something that's going to take time to enroll. It's in 13 countries and actively looking for participants. So the third ASO study I wanted to mention is the Curalis study. This is now in the entire population of people with ALS, because this is targeting TDP-483 related biology, where a smaller version of Stathmin-2 is made, and that leads to axonal damage. And so their drug is a splice switching ASO that makes full-length Stathmin-2. So now we're not blocking something, we're kind of making a full-length protein. So people are very excited about this because the science is very exciting. It's a multi-dose ascending dose study, so it's not a big study. They finished the first 2 cohorts of 8 people each and are now expanding. They think they have the right dose, expanding into a bigger cohort, cohort 3. So like the SOD1 ASO, there's 3 loading doses and then monthly doses in the CSF. It is a double-blind study. Unfortunately, it's not approved in the US, so we are seeing this a fair amount in ALS, where while we might have a lot more regulatory flexibility for market improvement, they're very tough on phase 1, and so we are seeing a lot of companies go to other countries for those phase 1. So this is in Canada, UK, and the EU. So now I'm going to switch away from the kind of gene modification approaches into why we thought it was kind of time to try a platform trial in ALS. This is a long list. I'm sure I've missed some drugs. On the left in red are all the recent trials that were negative. So we still have a challenge in sporadic disease, and I think that's driven a lot because of where the biology is and where our ability to subset people, kind of that question we had before about the heterogeneous features of the illness. But we do have a few in purple that have had some mixed results, some positive, not on the primary, but some positive results on secondaries, and those are mostly moving to phase 3 type studies. And then in the middle are all the active trials that are going on right now, with the ones in asterisks are enrolling. But again, looking at the pipeline, in about 2018 or 19, we started to seriously dive into whether a platform trial would make sense in ALS. And so this takes a village to do something like a platform trial. And it really takes having a consortium like Neil's with the sites and the knowledge about the disease, as well as company buy-in and the foundation buy-in. So, the advantages of a platform trial over a traditional are several. And one is that for the participants, there's a decreased chance of being assigned to placebo, because you share the data from the people in placebo between the different arms of the study. The time to getting your answers is about 50% faster, and that again is because you can go with fewer participants because you're sharing the placebo data, but also that every time you add a drug, it's like an amendment to a protocol rather than starting from scratch. And then it's, for those same reasons, about a third of the cost. So it's a good way to screen through drugs, and of course you can develop platform trials in many ways. They can be early phase, late phase, they can be very adaptive, but we decided to kind of stay in the phase 2 space and keep it simple initially. As I mentioned earlier, we really talked to lots of people before we got started to get buy-in from the community on this before we got started. So how does it work? So there's a common protocol, or what we call the master protocol, and all the regimens, all the drugs in there follow the rules of the master protocol. And so if you add a drug, they're all called regimens. The core is the common protocol, but you can have some things that are specific to that regimen as long as they're not taking away. So for example, if a regimen wants to add a biomarker that makes sense for their drug, you can add that. Or if there's some safety thing that you have to add, you can do that. But basically the visit schedules and the assessments are largely the same. So we started in 2020 with 3 drugs. As I mentioned before, we made it like an RFP to be part of the platform trial, and we got 30 applications from different companies, and we picked 4, and 3 of them were ready at the same time, so we started with those 3. And then we added a 4th one, and to add that 4th one, it was literally a 9-day IRB review, and the FDA had asked us to wait 30 days, so it was about 30 days to add a new drug. And then we added another one in 2022, and then 2 more in 2023. And we have about 4 companies we're working with now that we're hoping to add in early 2025. So when we say regimen, it means the whole group, the active drug and the matching placebo. So it's, again, one protocol, there's a single IRB, there's kind of single central governance. And so far, we have 70 sites. We started with fewer, but we actually, about 50, we found that because the patient interest was so high in this, that our sites soon got kind of at capacity to enroll. So we added some more centers, and we've enrolled about 1,500 participants in it so far. So, for each regimen, we want to find out if the drug works, obviously, if it's safe, if it's efficacious. And so just to show what it looks like for one regimen, after they're screened for eligibility, they're randomized 3-to-1 to active drug or placebo. Of course, you can make this different randomization scheme, but this is what we picked to design it. And initially, in these first 7 drugs, we decided to do a 24-week placebo-controlled period and then followed by active drug. We are going to change that for the next 4 drugs. We're going to go to 9 months, and that's driven a little bit from Jefferson data as well as some of the data from our first 5 drugs. That again suggests we might need longer in ALS to see efficacy. The main outcome measure is looking at ALSFRS, taking into account survival. Again, in the 6-month study, there's, fortunately, most people survive. There's probably less than a 5% mortality rate. But the FDA still kind of requires this combined assessment of function and survival. We're also looking at safety and other measures of the disease. The other nice thing you can do in platform trials, and most trials, is to try to learn about the illness so that no matter what the outcome, if your drug works or not, that you're still adding to the field. So we've added quite a lot of good science to it. Everybody has whole genome sequencing done. We're doing neurofilament, obviously collecting blood and urine, CSF for other biomarkers. We built in some new digital apps to try to learn and be able to compare them to the more traditional outcome measures. So we started with speech and home, but every time we add a few regimens, we try to add something different based on what we're hearing from our NEALS members about interest. So in our next, for example, our next 4, we have, again, from a philanthropic gift, we're going to add collection of PBMCs for everybody so that we can make IPS lines. And that's very exploratory and something maybe pharma might not want to fund, but to see, again, as we get better models of the disease, whether we could use that as a way to see who was more likely to respond to the drug. So it's a bit exploratory, but it's something that you can do in these, again, kind of academic-led platform trials to try to learn about the disease. So when we started, we wanted the first 4 drugs that we picked, we wanted them to be on different targets, because in the 30 that applied, we did get some companies that were going after the same target, but we thought at the beginning we'd try to do different targets. We now, when companies apply, that's no longer a criteria as long as they have good science, because we figure if 2 companies are on the same target, they're going to compete anyhow, and if they're going to compete, it's better to compete in the platform trial where, again, you're sharing the placebo data, it's more efficient. So the first one, zilucoplan, worked on as a complement inhibitor. Second, verdiperstat worked on myeloperoxidase. It was supposed to block microglial activation. The third works on energy metabolism. The fourth was a sigma-1 receptor agonist. And the fifth worked in autophagy. And then the last 2 that we added do have the same target, and they work on the integrated stress response. So when someone comes into the platform trial, they're randomized to whatever options that are available at that time. So that is one of the big differences between a traditional trial and a platform trial, is the patient doesn't get to choose which trial they're participating in. But again, that's another reason why it's good to make sure you have a good therapy selection committee that's making sure that there's good science for every regimen that you pick. So, for example, at the beginning, people came in, were randomized to A, B, or C. Once D came, that could be an option too. And then more recently, it was really just F and G. Then there's a second randomization. Let's say someone's assigned to regimen A, and they'll know that they're assigned to regimen A. Then there's a 3-to-1 randomization of active versus placebo. But when regimen A, let's say, is done, then we take all the active drug from regimen A and we compare it to the shared placebo group from A through D. Now, we have a big placebo group now, but we have had lots of discussions with the FDA where they're still not completely comfortable with the idea of using what they call non-concurrent placebos. So for F and G, for example, we are comparing them only to the placebos of F and G because in time they're aligned, and A through E were before. Now we're going to go back as soon as we have the data to try to argue that which I think we can show from A through E, that there really isn't a placebo effect or a time effect change in ALS, but I think we have to prove it to them before they're going to really accept it. So it's not I think it's just part of what we're learning about platform trials. We like the idea of having a pool of placebos you can share, but right now the regulatory decision makers don't like that idea quite yet. So we have results from the first 5. 3 of them were clearly negative. Regimen A was actually stopped early for futility. I didn't mention that, but we do have interim looks every 12 weeks once a regimen has accrued a certain number of people that have finished 6 months. We wanted to do that, so that we don't waste people's time. And actually, by stopping regimen A early, we actually did save a lot of time and as well as funds for that. Verdipstat was just clearly negative on all the outcome measures. But again, the feedback from Biohaven to us was, we got, of course we wanted it to work, but we got a quick answer in a very efficient, way. That was clear. Regimen C and D both did not hit their primary outcome measure, but had positive results on secondary outcome measures. So clean had a survival benefit as well as effect on neurofilament. Perdopidine had an effect on our digital app on speech, on multiple characteristics of speech. Perdopidine is a little bit like Nuedexta, a probably more specific sigma-1 agonist, and also had an effect on neurofilament and function in people who were earlier and faster. So those were all pre-specified, but not the primary analysis. So Prolenio is going forward to phase 3. Clean is in discussions with the FDA about the next steps. Trehalose was definitely negative. So again, in the 2 or 3 year period, we got answers to 5 drugs, 3 of which were very clear, and the 2 are maybe not as clear, but going forward to another replication study. Regimen F and G are enrolled very fast, and we're expecting those results by the end of this year. So now we took all the learnings from this, and we went back and looked at the data from A through E, did a lot of modeling simulations, and that really led us to decisions about some modifications to the master protocol going forward. And those modifications are largely to go longer, to go 9 months, because especially in regimen C, some of the positive effects were seen more in the open label phase, like what I showed for tofersen, and also to change some of the inclusion criteria to kind of get not include people who have more of the slower forms of the illness. So it's always a balance there, but we wanted to take the learnings for the first 5 and modify it while we have a couple months where we're not adding another drug. But right now we're in discussions in design phase with 4 other companies. So we do think by early 2025, we'll be enrolling again in the next group of drugs. So as I mentioned, we've published the design as well as the statistics and the master protocol. The hope, if people are thinking about platform trials, that this could be helpful. We have this great kind of outreach to the patients to help. As I mentioned, and I don't have exact numbers, but these trials are enrolling about 3 times faster than traditional ALS trials. And I think that's largely because the patients felt they were part of this and that this is a very patient-centered trial. And the patients are talking to each other about trials, and they're grading trials, and they're kind of pointing people to go into what they think is a well-designed trial. So just to summarize, I hope I've convinced you that the treatment landscape is really exciting. There have been some successes. We have a lot still to learn about, particularly the singleton form of ALS and what the best biology is there. The ASO deliveries in the CSF have been shown to be effective, have really told us that this is a treatable disease. And I know that sounds silly, but I don't think we really knew that until we actually saw people stop progressing with SOD1 ALS. And we also learning that it might take time for a drug to work and to see that healing and that repair. And these, new trial innovations are exciting. I just showed you the one we're doing. There's platform trials now in UK and also in Scotland, and people are trying different ways to try to go through what is, again, a rich pipeline and try to get some answers for patients faster. So I just want to thank the Niels group, which is just an amazing group like you guys that has a lot of fun together and learnings together and shares resources and really trains the next generation together. My team at the Healy Center, and then all the groups that fund us. Thank you.
Thank you for that wonderful overview of the clinical trials and the platform, of course. One of the motivations for a lot of our patients to enroll in clinical trials is open label extension and access to drug. Does that in any way integrate into this model?
Is it integrated into the what?
Is that an opportunity in this model of platform clinical trials?
Yes. So every regimen, they have to provide the open label extension. And it is, you're right, the patients want that, but it's also very informative. I mean, we wouldn't have known that tofersen worked if it didn't have that open label extension. But so yeah, it's required. While I'm on the topic of that, it is required, but it's actually enormously expensive. The open label extension actually costs more than the randomized control part because people are on it for longer, and you have to do the exact same safety assessments. And anyway, so it is a great idea, but it sometimes can be, especially for small companies, can be a challenge to do it as well.
But if it's a pooled placebo group, then which placebo participants get what open label? I mean, how does that work?
Yeah, they stay. If you're assigned to, let's say, regimen A, you get regimen A open label. Again, that's because we're using that data to learn about the illness.
Are there enough SOD patients to satisfy the recruitment needs for all these promising trials?
I don't know. It's a great question. I think I know some. There's been a couple of thoughts about how to do it. One is that the patients with maybe the mutations that are slower might be more open to the idea of going into the newer, one-shot trial, with the idea that they can get rescue tofersen if they don't work. It might be hard for someone who's going very fast to do that, and then at least in Asia, the number one mutation is SOD1, as opposed to here where it's C9. So a lot of the companies are also going to Asia for these studies.
So Merritt, thanks for that wonderful lecture, which is totally fitting for a Griggs lecture.
Yeah.
I heard you give a talk in Dallas at Carol Cruzen. That was about a month before Relyvrio results came out. So what are some of the lessons we learned from Relyvrio, from your perspective?
Yeah. Thanks. I did remove all my Relyvrio slides. No, so I assume everybody knows, but the FDA had approved Relyvrio based on a single study that was designed as a phase 2 study. It was a small 6-month study. And then the repeat study that happened in Europe that was not required by the FDA, but was required by Europe, was negative. So Jeremy and Schefter and I wrote a nice editorial in the Annals about it, but we tried to make a couple points. One, it really could be that the drug doesn't work. We've seen lots of small phase 2 studies not repeat. But there were a lot of things that were done differently. So the phase 2 study that led to approval was only in the US. It was in rapid progressors, 25 sites contained. The repeat study went for a broader population, 13 or 14 countries. It was much bigger, and it was longer, and they had a huge dropout rate. So, and again, I don't know if that's the reason the drug didn't work, but it was a change in design that I think that's one lesson. I think that if you're trying to repeat in the future, you can go bigger, but probably keep the same population and same type of design. I think we don't know. I'm not prescribing it. They pulled it off the market, but I do think that the repeat study had flaws in it. 40 to 45% dropout rate is a problem in any trial. And we see that in ALS trials that are long. That was a 48-week study, is that they're just over time, if they're still progressing, even if it's slower, they give up hope.
Just have a quick question. Thank you for that amazing talk. Did you find was it challenging to have all the companies together to work on a single sort of platform trial, or was it, what were the challenges in bringing companies together for a single protocol?
It wasn't. It was actually kind of fun. I think, again, because we we vetted it with them before, so people knew what they were coming in for. And, 3 of the 4 were pretty small companies, and they were excited to be part of this. And they did, they got in the same room together, and it worked out. I think the only challenge would be, I'd say, when you have that many studies all ending at the same time and everybody wants their results first, that was a little challenging. There might be a reason the papers are only coming out now. It was a lot of data all at once. That was probably the biggest challenge. Yeah.
So amazing progress, amazing work, Mara. Just congratulations on all you're doing. And thank you for giving me a little bit of credit earlier in your career.
It's true.
Which
Very
I
true.
probably don't deserve. But so now we are finding the causes of many of the cases of ALS, gradually picking them off. The strategy in the past has been just to treat all ALS patients because the disease is remarkably homogeneous despite all these differences in the genes. Do you think we should be investing mostly in finding more causes and going after specifically, like the SOD story is very impressive. We know the gene, we have a mark, we can follow it and look at how, but there's also lots and lots of patients that need treatment trials. So where do you put your money? Finding more causes and going after those individual causes, or taking the large number of patients we don't know the cause?
Yeah, it's a great question. I think it's maybe somewhere in the middle. I think that we do know that 98% of people have TDP-43 biology problems. I think the tools that we heard about from Pietro earlier, like if we could actually have either a blood or CSF biomarker that tells us that, that this biology is happening, and then we can have a treatment target of that, we might be able to, still treat the, larger populations. But I do think that the drugs don't fail because of the designs. They're failing because we don't know what to target. So I'd put more money that way, I guess.
Another question?
I just had a question about the inclusion criteria. A lot of our ALS patients have had symptoms more than the time frame of 48 months or whatever it's set to. And some of them are sort of slow progressors. So was there any thought about using either biomarkers or ALS-FRS trajectory to say, okay, maybe this person's had symptoms for 3 or 4 years, but they're really a slow progressor, and maybe it's worth putting them on a drug rather than a strict cutoff, looking more at how far along they are in their disease course in order to include more patients in the trials?
Yeah, that's a really good approach. So there's many ways to cut this. So I'd say that we've decided to try to eliminate slow progressors from our trials because in 6 or 9 months, they're just not contributing to the data. And you can do that with neurofilament now, actually, and as well as some other clinical characteristics. There's the NCALS group is doing it with a prediction model. It's called TRICALS prediction model, where they can put 8 different things and they can give you your prognosis. And so they're not having any cutoff other than the TRICALS score. So I think that is being tried, but in Europe right now.
Great, thank you very much.
Thank you.
Merritt, stay there.
Oh, am I staying up here? Coming down.
Yeah, so that was an absolutely superb lecture. Thank you for doing it. And this is a small gesture for you, which has got Dr. Griggs' name on it and your name. So congratulations. Quite heavy.
Thank you very much.
Okay. Yeah, just you. Great, congratulations.
Dinner. Okay. Dinner, and I think T-shirts.
Picture.
We're not done. Group photo. Okay. Group photo.
Yep.